So first up, we've got a bit of bioinformatics. Now, bioinformatics is a vast subject covering a large range of subject areas. So it's only going to be a taster of just the most important questions we tend to ask. Okay, so the headline for this session was what is out there? And the answer in bioinformatics terms is there's an awful lot out there. I mean, generally, of course, we're covering all of the usual sort of flow of information in biological investigations. So we cover the central dogma, of course. There's sequences. There's also loads of other molecules out there. This is all kind of in more general scope bioinformatics. And then, of course, there's stuff that, other biophysics techniques sort of tell us about the function. That's all, in some sense considered bioinformatics. So bioinformatics these days has grown from being just sequences into all sorts of other stuff. There we go. People can see my beauty. Lovely. So, this is a vast topic. I'm only going to scratch the surface. But there are things that we tend to do day in, day out, and I'll go through some of those strategies. And I put this in so I don't forget that, bioinformatics isn't just, a human or the mouse we happen to be studying. It's all known organisms for which we have molecular data. Okay, it's a vast collection. So there's an awful lot out there, is the simple answer. Now, what is out there is stored in databases generally, unless you collect the data yourself or have a friendly collaborator. And I've listed some of the most important databases here. You can, replay this talk if you want a little bit of a hint about where to go. Also, you can talk to, other people who do this kind of work day in, day out about where to go. But this is just a little snapshot of most important ones. So most commonly used databases are things like the genomic databases. So Ensemble and the NCBI databases contain the sequences and sequence-related information. We'll have a more closer look at that. This has sort of superseded the idea of having the gene database. There was this thing called GenBank. It used to have all genes known to humankind. Yes, it still has that, but these days you only think in terms of a species, but it's still there. There are databases to do with sequencing, to do with transcription, epigenomics, or epigenetics, so that's stuff on top of the genome, like histone marks and regulation, and there are protein databases. Now, it's too much. This is just a reference slide. How do I know where to go to find out which database? Well, you can ask people, as I mentioned. There's helpful reviews of databases. So there's the NAR article that's published yearly that's a roundup of the things that you should be interested in. And also you can go to hub databases like Uniprot, and you'll see Uniprot and Ensemble, these hub databases coming up again and again and again. They're the central organization, particularly in Europe. USA tends to have separate ones where we can sort of see where they link out to. Oh yes, and of course there will be databases for things like NMR and mass spectrometry and all of the other sort of details of biophysics of this. So this is just the main stuff. Yes, and if you want, so yeah, use hub databases to link out to other more specialist databases. So if you have no idea what proteomics or expression databases, have a look in Uniprot. There are links out, you can follow them. These will be things that are well respected enough to get Uniprot to actually include them. There's still 2D gel databases hanging around for some reason. There we go. Okay, what is this sequence? That's a question often we have asked. And you'll be given some gene ID, some gene name, protein ID, or a bit of sequence, or even the whole sequence. You want to know what is it? What is it? Oh, I want to go for my ID. I want to look up in the database. And then there's a question, well, which database? There are several databases, so we have to think about which one we choose. Okay, and I'll give some tips in regard to that. And if we're lucky, we can go from our ID straight into the right database. However, from that, we've got the entry, we've got the annotation. It tells you what your sequence is. Okay, that would be lovely. Sometimes it doesn't quite work out. Or sometimes that link is a little bit tenuous because IDs, unfortunately, go out of date. They get deprecated, they're redundant. And so sometimes we have to think about using the latest or the primary ID. Fortunately, in places like UniProt, this mapping is pretty automatic, but I've had issues with, say, old proteomics data where the IDs are just out of date. So be aware that that can be a bit of an issue. We can, of course, search in a sequence database with the sequence, and I'll talk about the search tools that are available to you. That often gets you to the right place, but sometimes it gets you to a related protein. Particularly if you're using a smaller database, it might, of course, only get you close. But close in terms of finding a homolog might give you enough information to tell you basically what your protein is, what it does, if that is known. Okay, so in summary, let's think about which database we need. You've got basically the choice of, do I want a big comprehensive one, fine detail, that tends to be slow to search, or do I want a smaller one that's more focused, maybe with reduced redundancy, so where sequences are clustered. That might be good enough if I just want to find some sort of family-specific information. We can, of course, choose a particular group of organisms. Do I want to just search in fungi or yeasts or in a particular species only? There's no point searching in UniProt generally if you know it's in one specific organism. And yes, also don't forget that sometimes you have unnatural sequences. What is my sequence? I remember looking through, I think it was a salmon genome. Couldn't work out what this was with BLAST. It was a vector sequence that had been left in. Okay, and there's actually UniVec, which is a database of vector sequences. Okay, yes, as mentioned, we can use IDs to get to the right entry. We can search with keywords, but beware that can be ambiguous. So instead of finding my isotochrome, it might be, you know, cytochrome associated protein or whatever. So obviously that's a little bit more imprecise. And then we can search for sequences as mentioned. With sequence searches, remember that the information you can infer from a homolog is only as good as the homolog is close to your query. So the more sequence separation, the more evolutionary distance you have, the less valid or less accurate the information will be. One word of warning with this sort of thing, even just looking up cytochrome B in this case, I ended up with almost 2,000 entries. Okay, there's a lot of redundancy in some databases. This is actually being reduced in UniProt. So beware of redundancy. Try and choose the best entry, the reviewed entries, or restrict it down to a single proteome set, where basically everything is just there once. Okay, and then finding things with sequences, use one of the common tools. I provide the links here. BLAST is still very commonly used. It's very quick. If something's certainly in the database, just use BLAST. Why not? However, there are other more sensitive methods. And basically, as I go down this list, I'm dealing with things that are increasingly sensitive to finding more and more remote homologs, i.e. where the sequence similarity is weaker and weaker. There's SciBLAST, which you can try. It's almost as quick as BLAST. The idea is it uses iteration, which I'll explain in a moment. And there are also Markov chain-based methods like HOMO, which actually started off in the LMB. Some of the early versions were done here, I think in cell biology, and HH suite. Now, what HOMO offers is it offers the ability to search with profiles. And by a profile, I mean an alignment of sequences. And an alignment of sequences actually contains more information than a single sequence. And HH suite is interesting because it allows you to search profiles against profiles. And a profile may be something like an alignment of a protein domain or a specific protein ortholog family. So we've got many options of increasing sensitivity. Now, I mentioned iteration and profiles. So what I was talking about here is that if you have a query, we can do a database search, find some matches. This is kind of what Jackhammer and SciBLAST do. Those will be aligned, and from those a profile is generated that allows us to search again with that profile. So we're widening the sequences and making them more general for the family. And then you can repeat this whole process. You go from alignment to matches, make a new alignment match again. And so you can get an ever sort of improving sensitive alignment. All of these searches tend to report something called the E-value. Just point that out to you. So there's scores associated with finding sequences, and generally the bit score means the bigger the score, the better. However, be aware that with e-value, the smaller the e-value, the better. This is the expectation for the number of hits, the count for the number of hits you would get with a score that's at least as good as what you achieved. Okay, so smaller is better. That means effectively that it's less random if it has a score that's close to 0. Another thing to be aware with e-values is that it does depend on the database and considerably depends on the size of the sequence you're searching. If you're searching for, say, a small peptide of, I don't know, 50, you might well get some good hits with BLAST or whatever, but rubbish e-values. However, all the e-values will be rubbish because the chances of getting a random hit with a small sequence can be proportionally quite high. Doesn't mean that there's something wrong, it's just you have to be aware that you can't tell so easily with small sequences. If the sequence is highly repetitive, yes, it might well be random, but if there's some reason to think that it's good, well then you have to take that forward sensibly. Okay, next question. I've got a gene. What's its context? Where does it sit? What's around it? What's going on at the genome? Okay, so I mean to focus in on one bit of the chromosome, where the gene lies, and have a look at the gene structure and everything we know about that bit of DNA. Unfortunately, this is made very simple because all we have to do in this case is go to a genome browser site, or you can actually download your own if you're really keen, and just look at that chromosome's location. And the data will tend to be preloaded. And I like to go to Ensemble, but there are other options, as I mentioned. And so just what used to be, going back in the day, quite difficult, very easy these days. You just go online, have a look at the right spot, and then that spot will have all of the layers of information that you can get bored with on top. So we can look at not only the gene structure, we can look at where it lies with regard to the other genes, what the synteny is, what the clustering is. We can have a look at all the regulation or variation. These will be displayed as data tracks on top of basically the linear sequence, which you can then locate a particular arm of a particular chromosome. And this is highly customizable. It tends to be preloaded with lots of useful information, but some browsers will allow you to upload your own data tracks to splat them on top. Okay, with regard to which browsers, I like Ensemble. It's broken up into different sections, so it has data preloaded for different kinds of species. And the WashU browser is very pretty. Stephen, my colleague, likes that one. And there are also organism-specific ones. So if you're into, Pombe, Saccharomyces yeast, flies, Dictyostelium, the organism-specific database will tend to be well supported by the community and will tend to have the most accurate, up-to-date, and curated information. And they have genome browsers that look like any other. Something else I'll mention is that there's a desktop sort of standalone application called SeqMug, which is a genome browser, and you have to import all your own data there, but that is free of the internet. So if you want to just put something on the laptop and go and have a look at it without using anything online, that is an excellent tool to use. That was written by Simon Babram. And on top of the genome, to give context in terms of functionality, we tend to look at short read experiments, so like the expression, the transcripts, ChIP-seq, ATAC-seq, all the other seqs that have come out since I wrote this slide. There are a lot preloaded in the genome browsers, but there are databases like GEO and ENA and ENCODE where you can actually go and have a look at, say, histone trimethylation in a particular cell line. There's loads and loads of stuff there. And ENCODE is just a system of trying to make a highly consistent set of experiments to give you data tracks to tell you all about everything from the chromatin 3D structure to fine scale regulation with transcription factors and whatnot. Okay, now moving on. I've got a protein. I'd like to know how it is organized. It's a deliberately ambiguous question. And so by organized, I mean, where are the helices, the domains, what's the fold? Tell me all about how this protein is structured and arranged. So generally the idea is that we will make our search more powerful by not only including the particular protein of interest, but we'll also include close homologs. The idea is usually we're looking at sequences that are close related, that share the same structure and set of domains. That makes many searches more powerful. Then typically the first thing somebody does when they look at their protein sequences, they say, well, what domains have I got? And that means looking at domain databases, and these databases actually have profiles of domains. So if an SH2 domain looks like this, okay, a kinase domain looks like that, and that's just a sequence-based search, and that will tell you what the domain organization probably is. It only tends to work for things we already know about, but I'll come to that in a moment. Of course, these days we can tell an awful lot quite accurately with things like AlphaFold to predict the structure. Generally, though, AlphaFold doesn't work on massive sequences, so we tend to run AlphaFold on individual domains that we think fold. However, if you can run AlphaFold on a lot, well, why not? That's the ultimate domain detection tool in a way. Otherwise, if you can't find something with domain databases, which things you've seen before, or structure prediction, which ultimately AlphaFold does use close homolog sequences. And so if there's not a lot in there, you might not be able to tell. Obviously, of course, that gives us the fold. Then you can still have a look at secondary structure prediction. These are quite accurate these days in the advent of neural networks. Obviously a bit redundant with full 3D structure, but it still can tell you what's folded and what's not, give you hints of what the architectures might be, what are transient things. And also something that people forget is that if you're trying to work out what the protein's doing, if it's conserved, it tends by it's doing something functional, and it's a functional domain. Yeah, you can spot this just by measuring the conservation values. Things that are unimportant, linkers, termini, that are not important, tend not to be conserved. So you can get some basic functional organization just by looking at conservation, which you get from aligning sequences, and I will mention that in a moment. Okay, so for finding domains, I recommend having a look at Interpro. This has absorbed what you might have heard of before if you're an old fogey like me, something called PFAM. So there's one big protein domain database. This is just Markov models to actually give statistics about how often you see a particular residue in a particular position. There's the PDB structures of those in a PDB subset. And something else I like to use is ProSite. It's got a little nice tool for building cartoons of your domain structure. So you can sort of play with those, bring up all the known information, basically. With regard to 3D structure, don't forget there are existing known PDB structures are in the database. Oh, excuse me. Okay, we don't just run AlphaFold. We don't have to. AlphaFold can be run from, well, you can run it yourself. There's ColabFold and other sites available on the internet. However, there are pre-calculated structures. So there's the protein structure database at the EBI, and also the Unipron contains links to, I think, probably the same entries. So a lot of AlphaFold stuff is already run for you. You do not have to run it. Okay. However, you really have to run it. There's things like ColabFold. I mentioned secondary structure. There's two great programs there. The last one, S4 Pride, recently updated. It's David Jones's one. It's very good. And so these are good for predicting domains as well as mentioned. And if you're interested in classifying your folds, there's Catherine Scott coming from the LMB. Okay. How does my protein or gene evolve? So we're getting towards thinking about alignments and things here. Now, the basic principle of how my biomolecule evolved is to gather a family. I want orthologs, homologs, paralogs. I want to look in different species to see what that protein has been doing in terms of evolution or gene. However, if we go back and look at different species, that's looking at ancient historical changes. That might not be what I want to do. I might actually want to look at recent changes. Of course, this is appropriate if I'm looking at viruses in the field or whatever. Okay. Either way, the idea is that we construct a multiple sequence alignment. Multiple sequence alignments are fairly tricky things to create actually mathematically is ultimately a gap optimization problem, but there's some very good software I mentioned that can kind of solve this fairly promptly for us. So once we've got a multiple alignment of our sequences, we can immediately see how the conservation of different amino acid sites or nucleic acid sites varies. Okay, and that gives us important biological clues. And then we can have a look at, once we've got the alignment, have a look at also trees. So a phylogenetic tree, just to say, well, this is the arrangement I think that my protein split and diversified in, might tell me something about how it is operating biologically. These are, this is a tree here of phosphatase regulated sequences I did for Anne, and you can see that there's some interesting paralogs going on to inform her stories. Okay. Right, so finding homologs in the first place is obviously the task we have to achieve. You can do all the sequence-based searches I've mentioned. That's a great way of doing it. Or you can use the pre-computed databases of orthologs. Why not? It's quicker. They've been checked. I really like the Panther database. Okay, but there are others you can choose. And this has got everything already clustered for you, so you can go in straight for your family and look at the important bits. As mentioned, remember whether you want to follow DNA or protein, if I'm looking at some recent variation, if things are changing fast or slow. And also, going back to the discussion of domains, you want to think, well, what am I studying the evolution of? Is it this whole protein? Is it just this domain? Am I tracking where domains get put in different situations, or is it just this particular thing? You have to make that choice. Anyhow, multiple alignments are generally required. My recommendation here is to use Clustal Omega, though muscle is also very good for protein sequences. And this will make your family profile and show your conservation. And there are online tools you can get to color these things in. And this is then the basis of the phylogenetic tree, usually after some cleanup. Now, for constructing trees, there's some good online workflows. Just plug in your sequences or alignments, press a few buttons, it will make a reasonable tree for you. And sort of medium fast methods like PhiML, it's a maximum likelihood method. This used to be slow on old computers, but these days it's pretty nippy actually, are really good estimators. However, if you want the gold standard for phylogenetic trees, it's still Mr. Bayes. This is really slow, Markov chain, Monte Carlo, and it's really fiddly, but it is, because it's really fiddly, it's very detailed and you can get very detailed information out of it. And at the end, of course, we want to draw our trees. Unfortunately, Mr. Bayes doesn't draw nice trees, but you can use a program like Dendroscope to make those lovely things like I showed you in the previous slide. Okay. Oh, yes, a little bit of vocab here. Some of you may already know this. I often use the word ortholog. By ortholog, I mean different species having the same protein. So they have arisen from speciation events. So the different versions in human, mouse, Arabidopsis, whatever, of the same thing. Paralogs is different. That's when your genes duplicate. It's a bit of a fuzzy definition because you can get orthologs of paralogs, etc. But ortholog means the same thing as far as we can tell. Okay, here's the big question. What is the function of my protein? Now, obviously, you're going to be doing lots of interesting biophysics to fill in the details. However, there's a lot you can do with just the existing bioinformatics databases. And as before, when we have a query, we can expand that by saying, well, what's just the function of this single sequence? What's the function of a bunch of homologs or orthologs, things that we think are doing the same thing in different species? And quite often, we can simply go to the database entries and look it up in Uniprot or whatever, and that'll tell us it does this. Well, that's great. However, that's a bit hit and miss. To improve things a bit, there's a system called the gene ontology, or GO. And the idea with that is we put annotations on proteins and genes in a regulated manner. So if something is called, a kinase, you know precisely what chemical reaction it relates to. And all the names of the organelles are specified. So this is a restricted vocabulary, if you like, and it's hierarchical. You can specify more or less precise information. It's still a bit patchy, but it's increasingly added automatically with sort of mass screenings and things. So there's an awful lot of information in Go terms. And because it's in a regular format, okay, it's easy to access. Now, if we don't have a direct link from database entries, we can also think, well, this protein is not acting alone. What's it interacting with? What pathway is it in? This can tell me about what function of that particular protein is. So we can look at interaction and pathway databases, get the partners of that protein or gene, and then that tells you more annotations about this. So we can enrich not only with orthologs, but also with interactions and connections. And then the idea is that, yeah, you might have different precise molecular functions in your pathway, but they have a common biological function. Okay. Now, one other thing that I've been looking to specifically is that when you can't get any further than the existing sort of connections in this sense, what else can you do with bioinformatics? One thing you can do is you can look at different tissues, different species, different organelles, using the principle that proteins that tend to act together tend to be present together in the same place, same time, and in similar amounts. So if you've got a gene, I don't know what it does, but it's only expressed in the eye, well, it gives you a clue, doesn't it? Okay. Here's a little example of that. This is a phylogenetic profile. This is a tree of species, and it just shows you whether certain members of a particular complex are present or absent. And so you can see actually that these are grouping together, doing a similar thing, they're in the same complex. So similarity of profile gives you a hint that these things are acting together. Okay. So basically what I've said is we can connect the annotation to annotations, so descriptions, including via orthologs, and the gene ontology is your friend. If you've got a bunch of proteins, say, that come out of some screen, you can look at enrichment of those gene ontology terms. You can say, what is special about these annotations? Okay, do they relate to, nuclear transport or, chromosomal modification or cell signaling in general? And so that's enrichment analysis is good for looking at clusters of proteins. Now, something that people often neglect these days are looking at the vast amount of medical information. So what does a gene or a sequence do? Well, what happens when it goes wrong? Now, a lot of information from veterinary and medical studies that show that particular genetic changes cause particular syndromes, particular conditions. Have a look in OMIM, and there's an animal version too, because it will give you a big hint about what a particular sequence does. Okay, so we can connect proteins and get combined information. Here are my recommendations for looking at the known interactions, biogrid and string and intact. My wife, who's also a bioinformatician, she loves string. That's pretty good. So the only warning with the known interactions, there tend to be a lot of false positives. People are overly optimistic, but it's good for restricting the field nonetheless. If you can, try and place your protein in a known pathway. Yeah, make sure you know that. That's pretty obvious, but those databases are out there. Oh yes, a little advert for my stuff. Not everything is known. Some proteins we know an awful lot about. This is a protein with a lot of Go terms. These are Go terms. Some proteins we know almost nothing about. Indeed, there's about, I don't know, 12, 15% of human genome we don't know anything much about at all. Okay, and I've got a little database that calculates precisely how much we do not know about certain genes. These might be interesting because they're not studied, and this database actually focuses on things that are conserved in flies, humans, etc. So if they've conserved, they probably are important. Okay, next question. Where can I get genome-wide data? Well, yes, you can download a genome sequence, but sometimes you want to customize it. And for genomic data, that's actually fairly simple. You can use Biomart. The idea is, in Ensemble, you can take your favorite genome and just get the bits of data you want out. So for example, maybe you want transcription start sites, gene lengths, okay, gene names, Ensemble IDs. whatever you want, just as a table. So you can put into a spreadsheet, do some coding with whatever. And it's very simple through that interface. For protein data, you can do a similar thing through UniProt. And generally, of course, you'll be choosing a whole protein, sorry, a whole proteome for one organism. And so there are loads of proteomes, and they're checked in UniProt. Be aware, though, that different proteomes have different levels of completeness. Buzzcode is listed at UniProt, and it checks how many of the well-conserved, constitutive basal machinery type proteins are actually represented in the proteome. So be aware that some proteomes are not necessarily complete. Anyhow, assuming we've got a good proteome, we can use UniProt to select particular genes of proteins and genes of interest, and then get whatever columns of data we want out. If you see it on the UniProt website, you can download it, basically. Okay, and yeah, this can be genome or proteome wide. All right, finishing up a bit. So that's a brief overview of some common questions, but if you've got anything detailed, you'll need help if you're not an expert. And there are loads of friendly bioinformaticians, people who dabble in bioinformatics at the LMB. There's myself. I often help people with protein sequence analysis, things to do with statistics and machine learning. However, if you're interested in DNA sequence analysis and processing, so the result of what Kim's going to talk about next, there's Stephen in cell biology and Alistair, PNAC. They can help you with, how to convert your DNA sequence reads into something more useful. And we're actually in the process of setting up a bioinformatics collective. That's basically a website where people lend their skills, or at least say, I'm willing to help people every Thursday or whatever. We also offer courses. So if you want to improve your programming skills, how to operate tools on the command line, or the cluster, sequence processing as mentioned, and also machine learning. I'm going to start a new machine learning course probably in January. Then you can sign up for that. It's open to all within the building. I include this slide just to show you where you can get some of the tools I've been mentioning. And this last slide I'll leave you with is where you can book all of the courses for programming. So for example, Stephen's got Getting Started with Linux and the Computer Plus coming up. You can book that if you want. I run Python programming and machine learning courses. I think Nick et al. Will be running some microscopy courses. So you can learn an awful lot, including an awful lot of bioinformatics through that site. Just look out for the emails when we advertise. Okay, I think I'll hand over now. Thank you.
All right, thanks, Tim, for leading into this. I'm going to talk about next generation sequencing, and I'm going to focus on Illumina sequencing. So this machine, the NextSeq 2000, was acquired at the LMB in 2022, and now we have two of them. And actually, if you think about the history of this place and Watson and Crick and DNA, and then Sanger as well, we actually don't do that much next generation sequencing at the LMB. So hopefully that's something that can be changed. And if you think about, I guess this is basically because a lot of structural biology goes on, which is fine. But hopefully this is a bit more of an introduction to what can be done with Illumina next generation sequencing and others, if you would be interested in investigating, I guess, outside the LMB a little bit more. So yes, I'll give a quick introduction. I'm going to try and keep this relatively basic. So those of you who have sort of learned about this before or been exposed to it before, it might be a little bit tedious, but bear with me. Hopefully I can still give you some insights. The diversity of sort of people's ability with Illumina sequencing is quite diverse at the LMB, I've found. So I'll give you an overview of how it works. I'll give applications or experiments that have been conducted at the LMB. And because I'm going to be unfortunately leaving somewhat soon, I will give contacts for people who hopefully will be willing to talk to you about those experiments should you want to do them yourself, okay? And then a few quick considerations in the first instance, and then a little bit more of other things that exist. Okay, so what is next generation sequencing? I'm going to put Sanger sequencing first. Okay, it's this thing that it sort of is not next generation sequencing, but there's no doubt that Sanger sequencing is still incredibly useful. Okay, if you're making any kind of plasmid or making any kind of DNA construct, The first thing, in my opinion, you should do at the end of the day is send it off for Sanger sequencing. It's really, really easy. And if it's not a thing that your lab is in the habit of doing, it's something that I would really, really recommend. It's very, very fast. It will basically give you data overnight. There's not that much analysis other than looking at it. It's also cheap from the point of view of if you submit only 10 samples, that will literally be cheaper than running an Illumina run. Okay, but next generation sequencing looks like this. Okay, so I think for me, it means that the sequencing is sort of highly parallel, meaning you can sequence many, many things at the same time, and you can sort of look at a single molecule. It's not necessarily strictly a single molecule sequencing technique, but you're generally looking at single entities, which means you can count molecules in a way that you cannot do with Sanger sequencing. Okay, and that will be sort of important later. So Illumina is the one that hopefully everybody is very, very aware of. It sort of started as Solexa in Cambridge. And it is, I think, generically short read sequencing. Okay, and now that actually Illumina's patents have expired, there are increasing numbers of competitors which are providing sort of equally powerful sequencing methods out there. So Elements is a particular favorite of mine. I think that I want to investigate further. But anyway, so Illumina has this. I think that's a relatively well-known picture where you basically make an array and you put your DNA on the array, and you're effectively imaging single nucleotides being attached to pieces of DNA. Right. Quite distinct is nanopore sequencing, which uses these nanopores, and you basically drag DNA through the pore, and as the DNA goes through the pore, you see differences in voltages, and those voltages are called as different bases. Okay, you might have also recently heard that Roche is releasing a thing called sequencing by expansion. This is effectively a technique to make nanopore a lot more accurate than it currently is. Okay. PacBio is something that goes sort of in between. It's single molecule real-time sequencing. It sort of allows you to do long reads, but using sort of more of a sort of polymerase-based logic as well. It has some advantages over both Illumina and Oxford, which I'll try and point out in a second. Okay, so Illumina is ubiquitous, meaning everybody is doing it, and there are lots and lots of tools out there, and so it's sort of the first place that you want to look. It is also fast, and it's very, very deep, which means it is statistically powerful. And so sort of in medicine, if you're looking for low incidence mutations, there's sort of nothing better than Illumina at this point in time. Okay. It also, if you're doing things like RNA sequencing, it has statistical power as well, right? Or CRISPR screening as well, which the others don't necessarily have. The disadvantage is that this thing is going to have relatively short reads compared to the other two methods, but the error is also relatively low. Okay. Nanopore gets you really, really long reads. So if you want to see long contiguous pieces of your genome up to sort of 1 megabase, I think a couple of colleagues have managed 2 megabases in one single read. This is better for repetitive regions, which struggle with analysis, and also structured regions, which form otherwise weird structures, which will then struggle to get through the Illumina sequencing platform. It's also cheap in principle, and also you can do modified bases, and given how small the thing is, it also lends itself to this idea that it can be done in the field. Okay? But so today that's not really possible, but it's something I think that they aspire for. It does, however, have very high error rates, which there are ways you can get around this problem, and it doesn't have so much depth. But generally speaking, Oxford, the nanopore sequencing will give you many, many errors. So if you're not sequencing the same thing multiple times, you tend to basically accumulate a lot of errors in your sequencing, which again, depending on the function, can be fine. And then PacBio compromises both. You can get these relatively long 20 kilobase reads. It's very important for if you want to do sort of the proper telomere to telomere sequencing of human genomes, for example. But it is expensive. It is also uncommon. It has now got a very, very low error because of a new technique that they've recently pioneered called hi-fi sequencing. But again, basically they're sequencing again and again and again in the rolling circle to try and get a consensus. Okay, so PacBio is basically not that available to the LMB. Okay, so if you're interested in it, we can have a chat about it, but I can and then point you in the right direction. But Illumina we definitely have, and that's quite ubiquitous, like I say. And Oxford Nanopore is also, we do have some too in-house, but I don't sort of run those as core facilities or sort of as a service for most of the LMB at this point. But hopefully, if you want it, then there's still the opportunity to do it. Okay, I'm going to go through as quickly as possible sort of the logic of Illumina sequencing prep, okay, given that it's the most likely thing that anybody here is going to want to do. Okay, it always starts with fragmented DNA, given that we have we can only sequence relatively short pieces. Okay, so often the first thing that has to happen is you need to isolate your DNA or RNA, and it has to be made small in some way, shape, or form. Okay, so that can either be done by sonication or by tagmentation or by fragmentation by an enzyme, etc. But the point is these DNA pieces have to be small. What then happens is you put adapters onto the end to basically label the end so that you can do more manipulation. And then the logic is because these things are going to anneal to a flow cell, you then need to attach in the process of library prep, the ends known as P7 and P5 so that the piece of DNA that you're going to sequence can find the flow cell effectively later. Okay, but there's a lot of varieties of what this looks like, which I'll hopefully show you in a slide or two. But the logic of all of them sort of follows this sort of trajectory. Okay. I've shown you these two priming regions, which I'll explain later. The barcode allows you to distinguish different pieces of DNA between different experiments or different samples. You can, if you think about how this works, skip if you know what DNA you have, which is somewhat common occurrence. Like if you're doing a CRISPR screening experiment, you often know exactly what your insert is going to look like. You can then directly PCR straight to this step. Okay. Common issues with this step, okay, sort of the biggest challenge in doing Illumina sequencing, generally speaking, I would say, is acquiring good quality DNA or RNA. If the DNA is good and has good QC, RNA likewise, effectively all the existing library prep methods will work really, really well. To be sort of good at library prep means you can do it on poor quality DNA and RNA. Okay, so but if you're designing your experiment from scratch, the first thing to think about is, can I confidently get a decent amount, meaning sort of 100 nanograms of good quality RNA or DNA? Okay, if not, you can still do things, but that's a really, really good place to be. Another common issue as you keep doing this more and more is you find these things called adapter diamonds. And that's basically the adapters stick to each other and they don't stick to the DNA that you're trying to get. That usually means you put too much of it in, but there are some ways to get around it, but watch out for that if you're doing this often. Okay, so I just want to show you this flow chart that I have for the logic of any library prep, basically. Right, and you can see on the left side, I have the start of your, these sort of the input. Okay, so that can either be whole genomes, whole plasmids, whole constructs, if you like, with no fragmentation. It can also be enriched DNA, and by that I mean if you've used an antibody, for example, to capture pieces of DNA, or you can be doing PCR to try and see how many copies of something that you're trying to investigate. And of course, you can also have RNA, and the RNA can either come from sort of extracting from a bulk sample, meaning normal RNA-seq, but you can also do all sorts of other things to your cells or to whatever sample you have in order to prepare it in advance. And then you can see in this first set of sort of processes, it's all to do with trying to break down the DNA into something that's more manageable for the library prep to deal with. And it can then be enriched after that if you would like to, so it can go back to the enriched DNA. And then the RNA, of course, needs to be reverse transcribed. Okay. So all of this needs to happen, but then all of this basically goes into this step, adapter ligation, which is what I was showing on the previous slide. Okay, the majority of applications are very, very good with an NEB kit called NEB Ultra 2. Okay, so again, if you're looking to do this for the first time, if you want a place to start investigating how this stuff works, they also have really, really good documentation and a really, really helpful tech support. So if you want to have discussions with somebody about how to design something more bespoke, they'd actually be happy to help you there. But the logic of most things goes through the NEB kits Ultra 2. And then there's usually a PCR enrichment. It's not necessary. You can do PCR-free library preps as well. But generally speaking, it just makes everything a little bit cleaner and a little bit easier. Okay. I sort of said here that this is another input, sort of skips a lot of the stuff, is if you know what, this is the PCR step, right? That gets you straight into the libraries that you need. Okay. This is what happens on the machine. Okay, so this is what a flow cell looks like for the NextSeq 2000. It is, if you zoomed in, you would see basically lots of these sort of nano wells, which contain a lot of these P7, P5 adapters, which would then connect basically that piece of DNA that I showed you that we were appending to the ends. Okay, and what happens first is that, so you can see the P7, P5 attached to the ends of these oligos that are on the flow cell. And then you do an enrichment cluster amplification. And basically the idea of Illumina sequencing, I'm sure as a lot of you know, is you're going to image these fluorescent nucleotides, right? The point of the cluster amplification means that you amplify that signal so that you can actually see the light in one image, basically across the entire flow cell. Okay. You can assess the quality by basically seeing of this clustering process. And again, this is sort of where the run is likely to not work, is if your well is empty and has nothing in it, so it will be black, or if a machine detects multiple colors in the same well, and that basically means you have multiple copies of the DNA in it, and then all of this is going to lead to decreased quality of your reading, okay? So the common issue is over-clustering, which means if in doubt, you should always load less DNA, for those of you who might have done this before. Okay, Illumina tech support is pretty good for discussing this kind of issue, but again, talking to other people who've done this before is also a nice thing to do, okay? This is the sequencing by synthesis cycle, okay? If you've seen these kits, they often label the number of cycles associated with the sequencing run itself, okay? And basically it looks something like this. The nucleotide itself is fluorescently labeled and has a reversible terminator attached to it. That basically means it follows the same logic as Sanger sequencing, meaning it will effectively stop. In one cycle, you add one base, you image the base, and then you remove the terminator and the fluorescence at the same time before adding the next one. Okay, and so basically it will do this for every single one of those nanowells that we saw before and image every single cycle and then compose those images together in order to give you the sequencing output. Okay, and this can be done also with the barcode as well. So the barcodes and the reads themselves are sort of primed separately, and so that's how you separate the barcode from the region that you're actually trying to read. Okay. I'll just quickly show you that this is how the sort of chemistry works in terms of fluorescence. Actually, there's only 2 colors instead of 4. Okay, originally it was 4 colors, now we only use 2. And you can see that A is basically both green and blue at the same time, and G is actually nothing. Okay, so this indicates actually that if you were to load G-rich sequences into the beginning of your sequences, actually it wouldn't detect those, and it would think that the well was empty. Okay, that's not actually a problem I've ever seen, but in principle it could be. Okay. This YouTube link at the bottom is a really nice video, which has basically these are just screenshots from that video. So it's a relatively nice thing that I made. Okay, XSleep is a thing that you might have seen in NextSeq documentation or things. XSleep is just new chemistry that was released very, very recently. It is faster and more and cheaper. I don't see any differences between it and the normal SBS, but both of them are still being sold. And my advocation is that you purchase the XSleep unless you know why you are purchasing the SBS, and so far I haven't noticed any difference. Okay, I'm going to very briefly go through also what analysis looks like. Okay, so lots of things happen on the machine. Well, 2 things happen on the machine already. One is that the images themselves are processed and turned into actual bases. Okay, and these are called base call files, which are these BCL files. On the machine, I usually advocate that those are then converted into the sort of the standard file for Illumina sequencing, which is called FASTQ. Okay, and that effectively converts all of these base call files into an actual sequence that you could actually read yourself if you wanted to open the FASTQ file in the terminal window, for example. It will also, if you offer the indexes that separate out your samples and your experiments in advance, also demultiplex, and that's the process of separating out which different indexes you have in this process. Okay, if not, though, that can be done. Also, all of this can be done off the machine, but it's much easier, I think, if you organize it so that it's done on the machine itself. Okay, the first step, almost certainly, of all of these things is to align these reads. Okay, so usually Illumina sequencing asks you to sequence small pieces of no more than 300 base pairs, which are from something which is usually thousands or millions of bases long. Right, so the process of identifying where that short 300 fragment goes across the whole genome is not necessarily trivial. There are a number of different aligners that you can use, different ones for RNA, also different ones if you are trying to align nanopore reads, for example. Okay, so this is a process that is well documented, but it is complex, and there are lots and lots of considerations in regard to which aligner you want to use. Okay, but what you can see there is I've sort of highlighted an entire read, right, in the box, aligned to the parent sequence. Okay, and so this is sort of a general genome viewer window, which sort of allows you to see where it is. Okay, and you get stacks of these reads on top of the genome that you've got. Okay, if you were doing things like ChIP-seq or RNA-seq, you would see peaks because you would be sequencing only the reads that arrive from at certain locations. Right, and those locations, that's why you often see with the genome papers, you would see peaks showing where antibodies bind or where RNAs that are particularly expressed are, et cetera, things like this. Okay, you can also see that I've got a G mutation. And so again, that's sort of part of the process. If you're trying to find consensus, for example, you can see how many times do you see mistakes in the sequencing? Okay, so after this, there are usually two things that happen. One is you do some counting. Okay, so again, the digital aspect of the sequencing is really important here. It's sort of what made, I think, the Illumina sequencing very, very powerful in the first stage, is you can count how many times you find, for example, a certain RNA. Right, so if you're comparing between two different conditions, you're effectively saying, well, RNA levels have increased given that you've added, you've added a drug to this system, for example. Okay, if you're doing a CRISPR knockout screen, the logic is which of my CRISPR gRNAs survive the process that you've applied in order to try and work out what your pathway or function is currently doing. Right, and again, it's important to do the counting in order to do this. With consensus basically means if you sequence a thing enough times, after a while, you'll be pretty confident exactly what that sequence is, even within the Illumina sequencing error, right? So for example, the Illumina sequencing error is 1 in 1,000. So it would have to sequence the same error, basically, 1,000 times in order to see that you've got basically, you can be confident that there is a mutation in your plasmid, for example, that you tried to put there or one that you didn't want to have there in the first place. Okay, so consensus is usually the other thing that happens. Okay, I've put this up as a quick summary of the Illumina experiments that I've sort of been aware of in the sort of last 3 years that this machine has been around, okay? Led with a few questions across the top, if those are things that you want to sequence, so sort of think about yourself. At the very end, is my DNA assembly successful? I think I found, and correct me if I'm wrong, I guess this might be outdated, but what I generally find is across the LMB, there are sort of quite a lot of attitudes towards what you do when you make plasmids, right? And I think going forward, given sort of advances in tech, for example, and using computers and things, it's nice to store full sequence data of all of your plasmids that you might want to make, right? It's an easy way to check that you've got the thing that you want, or that you haven't accidentally sort of disrupted a plasmid or a construct that you've been using for a long time, right? It's not that uncommon that somebody takes a plasmid that they've got from like 10 years ago, sequences, and finds actually like half the things have changed from the original, which is fine, and it might still work and do the thing that it wants, but generally speaking, I think it's nice to now have this kind of data. You can also do this with a service, right? If your plasmid is small enough, you can just send it to Source Bioscience, and that would also be fine in this case. But a lot of the synthetic genomics that you might know has happened in the LMB over the last 5 years or so is sort of heavily reliant on whole genome sequencing for consensus. Okay, more commonly though, I guess as biologists, you ask these kinds of questions. Often these days you find in papers, or it has been the case in the last 10 years or so, when you finish your experiment or you've made a new cell line, you want to characterize the thing, and often a paper would then say, can you just give me an RNA sequencing data set to just tell me more about this system, right, as an extra thing to add to a publication, right? So that's basically why I think characterizing the system is a very common thing that you might want to do these things for. It means that you only do one of these experiments sort of once, okay, once you finish most of the work, and it's sort of as a cherry on the cake, if you like. Right, but if you're looking for what things are actually important or relevant, for example, when you treat with a drug, or if you're trying to work out what's changing in between different conditions, if you're trying to work out if an antibody is going to bind differently to the genome under certain conditions, then these are things that can allow you to do this, okay? I mean, the two common ones are really at the top, right? One is the RNA sequencing, right, which is sort of very, very ubiquitous. There are like a number of sort of disadvantages to doing RNA sequencing, which are solved by nascent RNA sequencing, but nascent RNA sequencing is a lot more complicated, okay? The CRISPR knockout is also really, really popular. I have a pathway. I want to see which genes might be involved in that pathway or related to that pathway, okay? It's a thing that I'm asked to do quite a lot. And so this is the beginning of sort of thinking about how to design such an experiment. I will say at this point, I guess, that the CRISPR knockout screens requires you to have a good assay of your own. For example, flow cytometry, okay? So often you do flow cytometry to select the things that are sort of giving you fluorescence, and then you can then sequence the things associated with that fluorescence. That's often the design of such a thing, but they're not necessarily trivial, but they are sort of quite robust if you design a good assay for the thing. And then these are other things that have happened. So karyotyping is the Shabazi lab with Luca. ChIP-seq happens a lot in Andrew Mackenzie's lab and also in the ATV lab. Folks with Julian Sale, I can actually just put the next one up, sorry. Folks with Julian Sale and also in Joseph Yeel's lab do a bit of biophysics by sequencing, which is quite interesting, looking at sequence structure. Madeline Lancaster does a lot of single-cell sequencing, which I don't really look after because the NextSeq 2000 can't actually sequence deep enough to do most single-cell sequencing experiments, okay? And mutation rates is a thing that I've actually done, and also Julian Sayre is interested in as well. This is a thing that I think is for synthetic biologists, right? Directed evolution is a really powerful method to try and ask your protein or ask your system to do something, whether that's grow faster or to challenge it against an unnatural amino acid or a natural base pair, for example. But obviously sequencing is really important. Again, the counting, you count which sequences survive your selection process, okay? So what I've done here is I've listed a bunch of names that if you are interested in any of these things, I would advocate that you go and talk to them or their groups at this point in time, just try and think about whether or not these are things. So hopefully this slide will be available to you and you can come back to it and refer to what's going on, right? So again, it's just, I guess, ask nicely, but it's sort of try and utilize, actually, I think that the LMB is a really collaborative place, right? And so generally people are very, very happy to have coffee to sort of explain to you what they think is going on, right, for like an hour or so, right? And like I say, if I was still around next year, then I would basically be saying, just come and talk to me, and we'd have a chat about what interest you might have. And if I don't see you again, that's completely fine. And I'm sure they would feel similarly, or the group members that they ask you to talk to would also feel similarly about it as well. Okay. Good. There are, of course, loads of other Illumina sequences, experiments, out there. I have a huge poster at home of all of the sort of possible iterations, if you like, of Illumina experimentation. If there are things that you can find, the majority of the time, we can do them in-house on the NextSeq 2000, and if not, they can be done in other places, which I'll show you in a little bit. Okay. I'll give a quick list, again, this is like for reference, of things that you should think about when you design these things from scratch, right? So the first thing, like I said before, extract your RNA well, extract your DNA well. Okay, if you can get a lot of that, basically a lot of the rest of it will fall into place really, really straightforwardly. Okay, so I think both of these two kits you can get from stores, so they're like a good place to start. And they're pretty robust, I would say. Okay, the next thing to do is to QC your nucleic acid. Okay, that either is by using qubit high sensitivity or by using tape station reagents. Okay, again, I'm not going to go into the details of what you're looking for, but just think about these things when you start these experiments. How many reads do you need? These are the offerings that the NextSeq give you. Okay, so for example, an RNA sequencing experiment generally asks you for 25 to 30 million reads. Sort of looking at Stephen in case he wants to correct me and things like this. All right, so things will differ a little bit, but you can probably ask ChatGPT to give you a general idea of how many reads you want and then how many samples you have. Make sure you have replicates if you're doing biology as well. And therefore, you can sort of then have a think about whether or not this is enough. So for most RNA sequencing experiments of the kind that I've looked after, usually the 500 million read, £1,000 kit plus about 500 to £1,000 of extracting DNA and things. So about £2,000 experiment gives you an RNA sequencing experiment, for instance. Okay, these costs are they seem quite high, I think, against most of the other things that you do in the lab. But I can tell you for a fact that there are members of this institute which spend absolutely loads of money on sequencing. So the likelihood is, if you are doing any sequencing, it's not going to compare to the likes of Jason Chin or Madeline Lancaster. Okay, so you know, in the end, it is in principle the same pot of money, but I'm not trying to ask you to just like spend lavishly, right? But often people come to me and say, that seems really, really expensive, right? I'm really worried that this isn't going to work and it's a bit stressful, right? But actually that's the kind of thing that you should try not to think, I think, when doing these sequencing experiments, right? Okay, and then what happens afterwards? Try not to do all of this and then have loads and loads of data and have no idea what to do with it, right? And so again, I'm going to ask for Tim and for Stephen and for Mr. Crisp to help out with these kinds of things after I'm gone. Good. Okay, and then finally, CR UK across the road has the NovaSeq X, and that takes you up to 50 billion reads, right? So that's necessary for single cell sequencing, and also if you're looking for very, very, I doubt anybody will ever want to do this here at the LMB, is the sort of thing that Sanger likes doing a lot, right? Looking for very low instance mutations associated with disease, right? So that's where you can get the 50 billion reads. If you want to collaborate with Sanger and use their 26 of these NovaSeq X's, maybe you can do that as well, but I don't think that's happening either. But for single cell things, where I haven't talked about single cell and what it can give you, or anything like spatial transcriptomics, for example, these are not things I've had so much experience with, but these are very, very sort of exciting things at this point in time, particularly in development, actually. If you want to try and characterize a different cell type from a tissue, these are things that you can sort of consider. Okay, and Madeline Lancaster, again, I would really advocate talking to people in her group if you want to find out more about this process. Okay, in order to get access to CRUK, your group needs to have spoken to Christine Barrett to give you some cost codes and things like this as well. If you want to do nanopore, whole plasmid sequencing from Source Bioscience is super straightforward. It's something I actually recently tried for myself recently for the first time, and actually it works really, really well. Okay, you'll see actually if you look at the raw data how much errors you get from nanopore sequencing, but actually because it usually goes through maybe 10 to 20 times, actually you can be pretty sure that the sequence you've got is again this consensus, right? Julian Sale will have access to a MinION and a PromethION, which are the two sort of nanopore sequencing machines. And people in his lab can then show people how to use these as well. And we sent some samples off to UCL for PacBio, and they came back quite quickly. So if you really want to try PacBio, then UCL might be a good place to go as well. But I don't know too much more about that. I will also give a quick shout out to the robot in PNAC because it does do library preps. Okay, so if you are looking to do lots and lots of library preps, okay, so lots sort of means 100 or so regularly, so not just once, I think. But if you want to do, say, every month you want to do 100 or 200 samples, then using the robot to do library prep is quite a useful thing. Okay, so that's, again, not entirely sure who's going to be organizing that kind of thing after I leave, but I'm just telling you that there is a robot here and there are people who are keen to learn this thing, and it is possible to sort of just learn. So something to also think about, just be aware that there is robots. There's also robots in crystallography, I should say, as well. So Fabrice also has access to some other robots. Okay, so there's some ways you can think about doing high throughput experimentation. It's not traditionally a thing that the LMB has been sort of very interested in just by the nature of the work, which is, again, very, very good and well. But if you are interested in upscaling, then this is a way you can consider. Okay, the nice thing about this robot is it's connected to a bunch of things like a thermocycler, plate reader, incubator. So in principle, it can do a lot of experiments by itself. Okay, I'm going to finish. I have a bunch of acknowledgements. These are all the people I've done sequencing with, and it's been really, really fun learning about their experiments and sort of helping them out a little bit and stuff like this. And then these are the folks who've actually been very, very useful to me personally. All the bioinformaticians, thank you so much. And Ben, who probably none of you will know, but he left some time ago, but he was the person who really knew everything about how the flow cell actually worked and the Illumina sequencing works. Two folks for Illumina who were really, really helpful, and then also scientific computing, because there's a lot of that going on as well. Cool. Yes.
Hello, everybody. My name is Cat Franco, and I'm the head of the mass spectrometry facility here at the LMB. And obviously, today I'll be speaking a little bit about mass spectrometry. There we go. So I'm going to try and guide you through a ride of what we can do with mass spectrometry, especially proteomics within the LMB. Before we step into the really interesting things that we can collaborate and do together, I'm just going to give you very key subjects of what is mass spectrometry. So mass spectrometry measures mass to charge, not just mass. What we do is basically we look at the isotopic distribution of our analytes and determine the charge. And once we know that, it's a very simple calculation to understand the mass of your analyte. We do this frequently because we mostly work in proteomics downstairs in the mass spectrometry facility. We determine the mass of the peptides. We determine mass of the fragmentation of the peptides. We can determine the mass of intact proteins with or without their modifications. And we can, in some cases, do work with other biomolecules. And what does this type of information gives you? It gives you identity. It allows you to identify your analyte. And another thing that we can do, and I'll explain how we do this, is we can quantify your analytes. We can compare it across samples, and we can monitor how its abundance changes between your different samples, which then allows us to do some sort of correlation with the biological process that you are studying. So in terms of the mass spectrometer, it's a very complex instrument. I brought some teeny parts, and teeny because they can be quite big. Very complex instrument, but basically you can divide a mass spectrometry into three main components. The ionization source, which is, this is just an example, a very old example. We usually take parts from our old instruments to keep them for occasions like this. So this basically sits in the front of your mass spectrometer, and it bridges any technique that's siphonated to your mass spectrometer, which usually the case is an HPLC, to basically allow to drive your analytes inside the mass spectrometry. Usually in the front, it depends on the source. There's multiple ionization sources available. But usually in this case, which is electrospray ionization source, we have a needle It could be a metal needle, it could be other materials, but usually it's a conductive material. And if you imagine, then soon after you have the entry of the mass spectrometry, it kind of looks like this. So basically, this is what makes it a glorified vacuum cleaner. It's in vacuum inside, and all your analytes will go inside from this point of entry, which is a simple, basically, needle. Right, and then inside, there's multiple ion optics, what we call ion optics, that kind of look like this. This is also a mass analyzer, so this is a quadrupole, and it allows us to move the ions inside the mass spectrometer. Right, so we have the ionization source, we have the mass analyzer, which obviously it's where the analysis of your molecules will happen. It's where we'll determine the mass overcharge ratio. And then you have the detector, which basically will translate this information to something you can use, numbers. Right, something that's key importance, mass analyzers. I won't get into details about the different mass analyzers that we have and all of those that are available. I did do a lecture, I think it was last year, if not the year before, about mass analyzers. So if you're curious, you can just hop on to YouTube, exactly. The YouTube of the biophysical lectures, that's what I would say, the biophysical lectures, and have a look and see, or come down, and I am more than happy to talk to 2 hours about mass analyzers. Not the case though, I'm not going to do that. Right, so usually we have a mass spectrometer hyphenated to some sort of technique. And this basically is because the mass analyzers are not extremely fast. Nowadays, there are some pretty good fast analyzers out there, but it's not always the case, which means that you kind of have to give time for the mass analyzer to be able to analyze as many ions, as many molecules inside your sample as possible. And we usually do this with a separation technique, which is the HPLC, as you can see here. Let me just grab this. This is the point of entry. So this is basically a C18 reverse phase column where we inject the peptides. So they go through here. This could be 5 centimeters up to 60 centimeters. Obviously, the longer the columns are, the lengthier the grade it is, but it gives you more resolution. It gives you the power to separate more biomolecules inside your sample. And off they go inside the mass spec. And this part here is exactly this one. So this is the entry of the mass spec. And this is how it looks. So they start going in the mass spec, and this is the signal that each of these ions produces when it reaches the detector. And what we can do, we can identify, which I already told you, but we can also measure these intensities. And this allows us to quantify each specific of those mass overcharge elements that you're seeing. We can then compare between the different samples that you have or your different groups. And then we monitor how these abundances are changing within your groups. And this is how we can, for instance, see if there's any particular protein or a protein pathway that's having a relevant effect in your biological process. Now, about identification. Sometimes, or 20 years ago, we used to say, right, just by looking at the mass of a peptide, we can identify it. We know what it is. The reason why is because we usually do a trypsin digestion of your sample. We can also identify, or we can work with intact proteins, but to do standard quantitative proteomic analysis, we do chunk the proteins into smaller bits, peptides, which we know where they should be cleaved because we use trypsin. So it's either an arginine or a lysine terminated peptide. And the good thing is that nowadays, even though we have a great power in terms of the mass accuracy that we can get from these measurements, the community also realized there's far too many isobaric or having the same mass, peptides having the same mass, that looking at the intact mass does not give us the information of absolutely sure that what we have there is what we think it is. So we fragment these peptides. And because there's a multitude of different fragmentation techniques, but the cool thing about them is that they're predictable. So we know where peptides are going to be fragmented. And remember that we do see mass overcharge. However, what happens is that you have your peptide, and once you fragment, you get almost like a subtraction because we know it's going to fragment. We have a subtraction of each amino acid. So we can derive the sequence of a peptide just by measuring the masses of the fragments. And this is how we identify. Right, so I think I've given you all the points about mass spectrometry. I'm going to stop here. We can chat if you're curious about more, or have a look at the YouTube channel. There's lots of information. What I'm going to do now is going to a different point of view, which is, right, mass spec is really cool, but how does it work? How do I actually get it to work for me? What questions can you help me with? So let's imagine, we'll do this by sample type. I have a gel. I've been working on this complex, or I've been producing this protein, and I need to be absolutely sure that I have it. Well, that's one of the things that we can help you with. We do identification of the proteins. So the only thing you have to do is just excise that little gel bed, bring it to us, or you can do it yourself. We'll get there, and we'll do the identification. So we'll do the digestion, we'll inject it in the mass spec, we'll check the masses, we'll run the bioinformatics workflow, and then we'll let you know, right, it's not your protein. Quite common. We can also help you in some situations. Oh, I expressed my protein. I'm pretty sure the Western tells me that it's my protein, but it's not running in the molecular way that's supposed to be running. So what is this? We can let you know if it's modified, if it's a truncation. So we can try and help you if there's something strange going on with your protein, because anything that creates a protein changes its mass. So we can try and figure out what's the decoration, because we can see that there's an increment in mass. In some situations, this is easier said than done. And the reason why is because some of these post-translational modifications are really low in abundance, meaning that only 1% of your protein is going to have this modification, and all the rest, it won't be modified, or it will be modified with something else. In such situations, what we do is we do an enrichment. So it's kind of an affinity capture of the modified peptides that we can then validate, right, your protein is phosphorylated, your protein is glycosylated, or something else is going on with the protein and we have no idea what it is. That's also a possibility. Right, now let's change the sample type. You have a purified complex. You've been working with this for a very long time. You want to go cryo-EM because you want to see what's the structure of your purified complex. How can we help you? Well, first, we'll let you know if it is your complex or not, if all of your proteins are there. That's quite important. We can help you with the intact mass of your complex. So the intact mass does give a lot of information. It tells you, especially if you're working with a protein complex, is it expected? Do all your proteins have the mass that they should have? If you're working with a protein and a binder, is my binder there? We can do an intact native mass spectrometry analysis in which we won't denature your protein, just make sure that your binders or whatever you're looking into to making sure that's there is still interacted is there. We're not killing that connection. So we'll do a native analysis of your protein. We'll let you know about the intact mass. And then another very cool thing that Thomas will tell you about tomorrow is we can give you a little bit more information about what is interacting. And the information that we can help you with is at two different levels. By cross-linking mass spectrometry, we will let you know, right, peptide from protein A and this peptide from protein B, they are connected because we can see them linked in the mass spectrometer. So probably it's one of the part of the structure that's driving your interaction. Or by another technique, which is deuterium hydrogen exchange, what we can tell you is a little bit more information. We can tell you what are the amino acids from this protein that are driving the interaction. Now, the two key things and take-home messages is cross-linking mass spectrometry. It's easier. In terms of the mass spec, it's faster, especially if you're working with protein complexes. If you go to InCell, it's a whole completely different story. But if you're working with complexes, you can have this information relatively fast because of the resolution of the information that you're going to get. Hydrogen deuterium exchange, on the other hand, is it's time consuming. So you need to do several injections, because basically what you're doing is you're exchanging your hydrogens, your surface hydrogens with deuterium, which means that you have to do this as a time course. In such situations, if this is something that you would like to do, we would very much welcome you in the lab to work with us. We'll train you so that you can do your hydrogen deuterium exchange experiments, because obviously we have lots of things running. And an experiment of this sort is difficult for us to just completely dedicate ourselves, because we're working with multiple projects. But you're more than welcome to step in the mass spec lab. Right. Now, one of the things that I think it's quite important for me to reference here today is that some of our workflows, you can actually do it yourself. So the gel band analysis, it's a very straightforward protocol. It's available in our internal webpage, so you're more than welcome to just go there and download the protocol. And it's as simple as you extract your SDS page, you do reduction of the cysteines, you calculate the cysteines, make sure that nothing reoxidizes, and you add your trypsin, so your digestive enzyme. And then you submit the analysis request, and we'll let you know, right, today we have 2 samples, just come on over and inject it yourself. And the same thing if you're working with purified, if you're purifying your complexes and you just need to do a very fast QC. I just want to know if it's there. I want to know if my proteins are there. You can do it yourself instead of having to wait for the results for sometimes 2 weeks. I'm sorry. This is just because we're handling a huge amount of samples. You can just do it yourself. In the same way, you'll get your protein complex. We'll train you, so it's a very easy protocol that we optimize in the lab. So it's basically you do the digestion inside a tip. It takes literally 3 minutes. You submit the analysis, and then you come and run it. Now, usually this is around 10 minutes per sample inside the mass spec. It doesn't mean that we can give you the results in 10 minutes. It depends on what's on the queue. If there's hundreds of them, obviously, the mass spec needs to go through the amount of samples that are in there. Right. Protocols are available. If you have any questions, come and grab us anytime. Now, usually this is the face that I get when I say, come on, join us. You're going to do it yourself. This is going to be exciting. And everyone goes, I know that they clock so fast. Oh, I know that there's so many problems with mass specs. That's true. That's entirely true. They are difficult creatures. But we're there to help you. We're always in the mass spec room. We're there from 9. In some cases, Thomas likes to be there until very, very late. We're there. So we are going to train you. And in all situations, you'll be there injecting your samples and no one will be around you. So we're all working together. Right, so let's continue. I have an affinity pull down. What can I know about my sample? What can mass spectrometry help me with? Right. First, we can let you know are the interacting partners of your protein, favorite protein. And then we can let you know what are the interesting interacting partners. If we compare your pull down sample with your controls and let you know, right, this protein is there on your pull down, but it's not on your controls. But we can go to a little bit in depth of this analysis, not just the presence absence, but there will be lots of proteins that will be changing in abundance that might be interesting candidates for you too. So we'll let you know. If you have cells and tissues, pretty much what we can do is we can look at the whole proteome. The depth that we usually get nowadays with instruments that we have in-house is around 8,000 proteins per proteome. We can let you know how these protein abundance, each protein, how its abundance is changing across your different conditions, and you can have as many conditions as you like. We will change our workflows depending on the number of samples that you have to analyze. And then we'll run protein identification and a quantification workflow. We can also let you know, right, but are there signaling pathways also that are being very important in my biological process? We will do this for you as well. So we can do a phosphoproteome analysis and let you know if there's any signaling cascades that kind of jump within your biological process. And a good thing is that we have kind of support from beginning to end. We can take the samples from the point of sample preparation, but we'll also be in communication with you with emails back and forth, saying, right, this is your results. It doesn't finish with a report. It doesn't finish with a list of proteins. We will help you try and understand the results that you have. It's very critical that you have this level of support, because obviously, if you're working with identification of one protein, you don't need much support with that. But if you're looking at the abundance of 8,000 proteins, you probably will be better if you have some sort of support to watch the better workflow in order to answer your initial hypothesis. Right. And this that I've just said, this is again in our internal webpage. So if you have your sample ready and if you want to remind yourself, what can I do with my sample? You can just go on our internal webpage, have a look. If it doesn't answer your questions, I'm more than happy for you to come across, ask the questions you want. I'll be there. And if I'm not there, just send an email. We'll reach out to have a meeting. Right. Tips, things that are really important to prepare your samples. When you're working on a gel bed, it's really key that you are working on a clean surface. And by clean surface, what I say, no dust. We'll see all your keratins. Your jumper keratins, if it's wool or if it's something else. So use gloves, clean surfaces. Purified proteins, whenever you're doing IPs, affinity purities, the beads that you're using, they usually are the killer of your experiments. And the reason why is because they are usually very heavy and you vortex and then you start adding to your samples. And the last sample will have more beads than the beginning. So we usually say vortex every single time. It seems very simple, but obviously everyone is so busy that sometimes they just go, and then you have different conditions and nothing is comparable. So, and the same thing for the inputs. It's really key. If you want to do quantitative analysis, you do have to be quantitative from the beginning. So if you're going to do affinity purification, you have to make sure that your input is the same across all your conditions. Otherwise, we cannot help you because it has been skewed from the beginning. So there's a bias. Right, and cells. The best advice that I can give is wash your cells. There's usually lots of proteinaceous stuff in the medium. Get rid of it and then bring us our cells. We'll handle the rest. That's it. So cells, not much of an advice there. Right, one thing that we do get questioned a lot, how many samples should I bring you? And the most basic advice that I can give you, well, let's start with 3. But this is basically, I will only know in the end because this is dependent on the variance of your project and of your samples. If you're working with mouse models, you have much more variance from each animal if you're working with a cell culture that has been there for generations and generations. So usually the minimum is 3. I might go back to you, let's do this again with 6, because it's not enough. We have a big variance in your sample. How much protein? This is a moving target. The reason why is every single year, mass spectrometrists get more and more and more sensitive. 20 years ago, to do 8,000 proteome exploration, we'll probably need milligrams of protein. Nowadays in the facility, we're working on the micrograms. Minimum, I would say 20 because of the workflows that we use. But with current state-of-the-art mass spectrometers, you can do this at the single cell level. So it's a moving target. Hopefully, in the near future, we'll have a mass spectrometer which is sensitive enough to give you 8,000 proteins in a single cell. Workflow, I won't get into this now because we're almost reaching the time, but it's very simple. We have to extract your proteins, and for this we use several different buffers that allows us to extract. Sometimes they have detergents which we really don't like. We do then the reduction and alkylation of cysteines, as I already told you. We do the enzymatic digestion so that we get peptides and inject them. There's many variants to this workflow, and it can get as complex as your running hypothesis. So it really depends. The workflow, this is something on our side. We usually select it for you depending on the objectives of your project. This is who we are. So each one of us has slightly different technical expertise. So do reach out to us, and you'll probably be able to speak to slightly different elements of our team because we have different things that we work on. Right, and this is just almost the final slide, how we work with you. Well, first, if this is the first time you're going to do a project with us, it's really good that we have a startup meeting. We'll define what's the workflow. We'll understand what are the objectives of the project so that we know what's the best workflow that we're going to apply to your samples. And then you submit when you're ready, you go back to your lab, you prepare your samples. When you're ready to bring them down, just submit an analysis request that's available in our internal webpage. Bring us the samples, we'll take it from there. And then you can go in our internal webpage as well and look at the sample queue that we have. This will be very informative because it's a live queue. We use the same software to see what, or to work within your samples, because usually we are working with different projects at the same time. And then we'll prepare everything. We'll inject your samples, and you'll receive an email with a link to download the results and with a report. And again, this is the first communication about the results. The process is a back and forward, right? I want to understand this, but I don't understand how this is being done, or I need a slightly different analysis of my samples. This is the beginning of our collaboration. And that's it. Feel free to come on down. I'm very happy to show you around the facility. I'm very happy to, if you're interested in doing your own analysis of protein ID or identifying your protein complexes, I'm very happy to train you and show you how everything works. And thank you so much for your attention today.